Hello and welcome. From today I'm going to talk about the development of the nervous system. It's the most complicated topic of embryology and is very difficult to understand in first year MBBS, but still I will explain this topic as simple as possible, okay? See, the nervous system consists of brain, spinal cord, peripheral nervous system, autonomic nervous system and ganglia or ganglions. All these topics, normal anatomy, you already know that because I talked about these things in my earlier videos. If you have not seen that, it's your fault, not mine. Everything develops from the specialized surface ectoderm, which is known as neuroectoderm. Okay. Now, this neuroectoderm presents in between the primitive node and precaudal plate. What is primitive node? You already know that. What is precaudal plate? You already know that because these things I talked about in my earlier videos. Notochord also presents in this area. What is notochord? You already know that. Okay. Now, one by one, you have to understand that in phase 1, it is known as neurulation. What is this? Neurulation is actually a process of formation of neural tube from neural plate. Now, what is neural plate? Try to understand. In my earlier videos of embryology, I told you that embryo folding stage where the head fold forms, tail fold forms, and two lateral fold forms. Now, in the head fold, specifically in between precaudal plate and primitive node, there is a surface ectoderm. Now, the center part of the surface ectoderm, it gets thickened when at third week of intrauterine life. Now, along with this, a part of the notochord also helps to formation of the neural plate. Now, this neural plate is also known as medullary plate. You understand how the neural plate forms? Now, within few days, this neural plate grows and elongates craniocaudally. Suppose this is the cranium and this is the caudal. Okay, so craniocaudally from upward to downward. Now, it gets lengthy. Okay. Now, at the beginning of 4th week, elongated neural plate forms a midline depression. Okay, so it is so long that in the midline it gets depression area, which is known as neural groove, and the elevated margins are known as neural folds. Now, this junctional point in between the neural plate and the surface ectoderm forms a neural crest. Suppose this is the neural plate and this is the surface ectoderm. So the in between this gap area is known as neural crest. Gradually neural plate more elongates and the neural fold comes closer to each other and starts fusion in the midline for craniocaudal direction. Okay, and it looks like a tube-like structure, so we call it neural tube. One open end cranial neuropore, another open end caudal neuropore. Now, through this neuropore, amniotic fluid circulates. Around 25th day, this cranial neuropore closes. There is a opening of the tube of the cranial direction. This cranial neuropore closes at 25th day, and around the 28th day, caudal neuropore also closes. Okay. Now, this closing requires 20 somite stage. Now, what is somite? You already know that, and how this 20 somite stages requires, you will get to know when you are going to do MD in embryology. Okay. And around 28th day, caudal neuropore closes. Now, this closing requires 29 somite stages. After closing, this cranial neuropore becomes lamina terminalis, and after closing, this caudal neuropore becomes terminal ventricles. Okay. Now, in the phase 2, you are getting the fate of the neural tube. What happens to the neural tube? Suppose this is the neural tube. One part of the neural tube forms the prosencephalon, another part forms the mesencephalon, another part forms the rhombencephalon, and the remaining part forms spinal cord. Now, the prosencephalon divides itself into two parts, telencephalon, diencephalon. From the telencephalon, we are getting the cerebral hemisphere, right side, left side, basically cerebrum. Entire anatomy of cerebrum, you know that from my earlier anatomy videos. Okay. Diencephalon forms the optic vesicle, pineal gland, thalamus, hypothalamus, posterior pituitary. All these things anatomy, you already know that from my earlier videos. Mesencephalon forms midbrain. About the midbrain anatomy, you already know that from my earlier videos. Rhombencephalon divides itself into two parts. One is metencephalon, myelencephalon. Metencephalon forms pons and cerebellum. Pons anatomy, cerebellum anatomy, you know that from my earlier anatomy videos. Myelencephalon forms the medulla oblongata. What is medulla oblongata? Entire anatomy of this medulla oblongata, you will know that from my earlier anatomy videos. Okay. And the spinal cord, you already know that from my earlier anatomy videos. Now, cavity formation. Cavity means a gap area. So, the gap area in the telencephalon is actually known as lateral ventricles. So, the lateral ventricle, we have right side and left side. This is also known as first and second ventricles. Okay. About the anatomy of lateral ventricle, you already know that. Cavity of the diencephalon is known as third ventricle. Third ventricle anatomy, you already know that. Communication in between the lateral ventricle and third ventricle is known as interventricular foramina of Monro. Cavity of rhombencephalon is known as fourth ventricle. About the fourth ventricle anatomy, you already know that. Cavity of mesencephalon, which connects with the third ventricle and fourth ventricle, is known as aqueduct of Sylvius. Okay. Cavity of spinal cord is known as terminal ventricle, which is also known as fifth ventricle. Fourth ventricle is also connects with the subarachnoid space via two foramen of Laschka and one foramen of Magendie. All these foramen, all these ventricles, you know from my earlier videos of anatomy. If you have not seen those videos, it's your fault, not mine. Fate of the notochord. Just now I said that a part of the notochord helps to form the neural plate, which ultimately converts into neural tube. Rest of the notochord, it helps to form the entire vertebra, dorsum celli of the sphenoid bone. Also, in between vertebra there is a cartilage, and in the cartilage you are getting the nucleus pulposus, which ultimately comes from the notochord. Okay. Now phase 3. In phase 3 you have to know that fate of the neural crest cells. Okay. What happens to these cells? During neurulation, the specialized cells presents in between the neural tube and the surface ectoderm. Okay. And it divides this thing into dorsal mass, ventral mass and others. Okay. Now the dorsal mass forms the neuroblast cells. These neuroblast cells forms the dorsal root of ganglia, sensory ganglia of the trigeminal nerve, facial nerve, glossopharyngeal nerve and vagus nerve. Skeletal elements of the pharyngeal arches, odontoblast of teeth, parafollicular cells of the thyroid gland. Now secondly, it forms the spongioblast cells. It forms the satellite cells of ganglion and Schwann cells. Pluripotent cells, melanocytes. Okay. About all these topics I told you in my earlier anatomy videos, except this Schwann cell and melanocyte, which I will talk about in my physiology and biochemistry chapter, not here. Ventral mass, it forms the sympathochromafin organ. Sympathoblasts, this forms the sympathetic and parasympathetic ganglionic neurons. Okay, you already know about these things. And the chromaffin cell, it forms the chromaffin cells of medulla of adrenal gland, then paraortic body, argentaffin cells of the respiratory system, enterochromaffin cells in the gut. All these things we'll talk about later. Okay, this neural crest cell forms facial bone, vault of the skull bones, muscles of the ciliary body, dermis of the face and neck, then smooth muscle of the blood vessels of face, forebrain, then sclera and choroids of the eyeball, substantia propria, cornea, pharyngeal arch cartilages, heart valves, pyometer, heart septums. All these things forms from these neural crest cells. Okay. Now up to this much today's video, I hope you will understand, and rest of the phases we'll talk about later. Okay. See you in the next video. Till then, bye.